Good afternoon, friends. Today we will discuss one of the very important concept in microbiology, that is bacterial growth curve. First of all, let us try to define the concept of growth. Growth is defined as increase in cellular constituents of organism. So when we talk of growth of any higher organism, we say that there occurs increase in cellular constituents, cell division, cell differentiation, all those terms considered. But when we talk of growth in microbiology, the growth refers to an increase in number of cells in a population. So concept of growth is different for unicellular organisms like bacteria. It is an increase in number of cells rather than increase in cellular constituents. So usually growth in microbiology occurs of a population. Therefore, it is frequently called as population growth. A population of microorganism grows because there occurs increase in number of cells of a population. One cell divides to form two cells divide to form four cells divide to form 8 cells. So in such a manner, cells increase in number. They are unicellular bacteria. The mechanism by which bacteria divide is called a binary fission. One cell divides to form two cells divide to form four cells. This division occurs by a process called binary fission. In higher multicellular organisms, we know that cell division occurs by mitosis. But in bacteria, there is no mitosis. There is no nucleus. So, cell divides by a process of binary fission. DNA replication is the first event that occurs during binary fission. Before a cell divides, the DNA replication must occur. Then only cell will divide by the process of binary fission, and then replicated DNA or replicated chromosome move to the opposite poles, and then cell increases the length. When a cell divides, all the components of bacterial cell increase in their number. For example, there occurs increase in components like ribosomes. There occur increase in number of plasmids. Whatever other cellular constituents are there in bacterial cell, all the components increase in their number. The time required for a cell to divide, to grow, and its population to double is called a generation time or doubling time. This is one of the important term in microbial growth. Generation time or doubling time, it is simply the time required for a cell to divide. Time required for one cell to form two cells. This is called a generation time or doubling time. Usually in unicellular organisms like bacteria, the generation time is very short. I can give you an example of E. coli. E. coli grows with a doubling time of around only 20 minutes in a nutritionally rich medium under optimal laboratory conditions. So only 20 minutes are required for one cell of E. coli to form two cells in a nutrition sufficient medium under suitable conditions. So they have very short generation time. Within 20 minutes E. coli can divide. Now, the different phases we will study in bacterial growth when a bacteria are grown in a batch culture. Remember, this batch culture is a closed culture system. It contains an initial limited amount of nutrients. For example, when we prepare nutrient broth and add a bacterial culture to it and allow this bacterial culture to grow for 24, 48 or 72 hours. That culture system is called a batch culture. In such a culture system, we do not add fresh nutrients nor withdraw the grown culture. Such a system is called a batch culture. So in this system, when bacteria are inoculated, they will go through different phases. And this growth curve is plotted by determining cell concentration with respect to time. So when we inoculate bacterial culture, they will multiply. The number of cells or cell concentration when it is plotted with respect to time, that is called a growth curve. Now we will study in detail different phases during bacterial growth curve or bacterial life cycle. First phase of bacterial growth is a lag phase. This is the initial phase where there is no cell division. There is no fixed time for lag phase. It can last for minutes, it can last for hours or even up to several days. It depends on the conditions. During this phase, there is no increase in cell number. Remember, cells do not multiply during lag phase. This lag phase is the phase of acclimatization. So when a bacterial culture is inoculated into a new fresh medium, bacterium requires certain time to get adjusted to changed conditions of environment, changed conditions of nutrient. So it tries to adjust to these conditions, and this time required for adjustment is nothing but a lag phase. During this phase, bacterial cells uptake nutrients from the environment. They increase in size, they do not divide. There occurs increase in cellular proteins, there occurs DNA synthesis, overall metabolic activity is going on, but no cell division occurs. Therefore, during lag phase, bacterial cells increase in size without division. So this is lag phase. In simple words, it is the phase of acclimatization or adjustment. Lag phase is followed by next phase of life cycle called logarithmic phase or log phase. It is also called an exponential phase. Once lag phase is completed, bacterium has adjusted to changed conditions. Since nutrients are plentiful, therefore bacterial cells will start to multiply very rapidly. They will uptake nutrients rapidly and grow very fast. During this phase, log phase, their growth rate is maximum. They will uptake nutrients and grow at their maximum specific growth rate because nutrients are plentiful. This phase is also called an exponential phase. Since this culture system is a batch culture system in which we do not add fresh nutrients, log phase doesn't last for a longer period. Because as bacteria multiply rapidly, their number increases, and this increased number of bacteria will lead to increased uptake of nutrients, and therefore since batch culture contains limited amount of nutrients, after a certain period of time the nutrients will start to deplete. Say after 2 days, 3 days or 4 days, The cell number when has reached to a great extent, definitely it has resulted in the depletion of nutrients. So what will happen, this microbial population does not maintain exponential growth for extended number of generation. Means log phase doesn't last for a very longer period. After a certain period of time there will be a plateau of the growth. One of the reason is essential nutrients get depleted, or other reason is that inhibitory products get accumulated. Or here we can say the number of growing cells is equals to the number of dead cells. Therefore there doesn't occur net increase in cell number as in log phase. In log phase there occurs very great increase in the cell number, but during stationary phase there is no net increase in cell number, and this could be the reasons that essential nutrients get depleted, or there may be accumulation of inhibitory products. The other reasons may be as a result of growth of the microorganism during log phase. The conditions in the culture medium may have got changed. For example, the changes in the pH may not be suitable, changes in the temperature may not be suitable for further growth of the microorganisms. The important thing to tell during stationary phase is that there is no net increase in cell number, and it may be due to either depletion of nutrients or inhibition of or accumulation of inhibitory products. That is called stationary phase. Now this most of the population in the stationary phase may simply cease to divide, although they remain metabolically active. It doesn't mean that microorganisms are dying. They are still active. The growth ceases, but the microorganisms are metabolically active. They are not dormant. So this is what happens in stationary phase. The last phase of cycle, bacterial growth curve, is decline or death phase. Here number of viable cells begin to decline, or you can say the number of dying cells is more than the number of growing cells. Now during this phase, the death of microbial population, like its growth during exponential phase, is usually logarithmic. That is, a constant proportion of cells die every hour. Means here number of cells dying is much more than the number of cells growing, because nutrients have been completely depleted. Other reason is accumulation of autolytic products. Cell causes lysis of itself. There may be number of reasons. The conditions become more and more unfavorable for bacterial growth. Nutrients get completely depleted. There may be number of reasons behind death phase. So number of viable cells begin to decline during death phase. So these are the different phases, 4 phases during life cycle of bacterium: lag phase, log phase, stationary phase, and death phase. This is the growth curve, which you can see. On x-axis there is a time, and on y-axis there is number of bacterial cells. Initial phase, you can see there is lag phase, no net increase in number of cells. Bacteria simply grow in size, they do not multiply. It is followed by log phase or an exponential phase, where there is a rapid increase in the cell number with respect to time, followed by stationary phase, which may be due to depletion of nutrients, and again the graph comes down with respect to time. That is called death phase. So these are the 4 different phases during bacterial life cycle, and this is called bacterial growth curve.